Mendel (1822 to 1884) was an Austrian monk who discovered the basic principles of heredity through his experiments. His experiments are the foundation of modern genetics. He was born in 1822 to a family of farmers in Silesian school of Czechoslovakia. After finishing his high school at 18, he entered the Augustinian monastery at Brunn as a priest. From there, he went to the University of Vienna for training in physics, mathematics, and natural science. Mendel returned to the monastery in 1854 and continued to work as a priest and teacher at high school. In his leisure time, he started his famous experiments on the garden pea plant. He conducted his experiments in the monastery for about 7 years from 1856 to 63. He worked on 10,000 pea plants of 34 different varieties. Mendel noted they differ from one another in many ways. Mendel had chosen 7 pairs of contrasting characters for his study. He chose the pea plant as it was advantageous for experimental work in many aspects. It is naturally self-pollinating and so is very easy to raise pure breeding individuals. It is short lifespan. It is an annual and so it was possible to follow several generations. It is easy to cross-pollinate. It has deeply defined contrasting characters. The flowers are bisexual crosses involving the inheritance of only one pair of contrasting characters are called monohybrid cross. For example, it is a cross from between two forms of a single trait, like a cross between tall and dwarf plant. Parental generation: pure breeding tall plant and a pure breeding dwarf plant. F1 generation: plants raised from the seeds of pure breeding parental cross. F1 generation were tall and monohybrids. F2 generation, the shelving of the F1 monohybrids resulted in tall and dwarf plants respectively in the ratio of 3:1. The actual number of tall and dwarf plants obtained by Mendel was 787 tall and 277 dwarf. External expression of a particular trait is known as phenotype, so the phenotype ratio is 3:1. In the F3 generation, 3 different types were obtained: tall homogeneous TT pure 1, tall heterogeneous TT2, dwarf heterohomozygous TT1. So the phenotypic ratio is 1:2:1, and genotype is the genetic constitution of an organism. Based on the observations, it was confirmed by Mendel that factors are passed from one generation to another, now referred to as genes. Tallness and dwarfness are determined by a pair of contrasting factors. Tall plant possesses a pair of factors represented by T, taking the first letter of the dominant character, and a plant is dwarf because it possesses factors of dwarfness represented as T recessive characters. These factors occur in pairs and may be alike as in pure breeding. Tall plants and dwarf plants TT. This is referred to as homozygous. If they are unlike TT, they are referred to as heterozygous. Two factors making up a pair of contrasting characters are called alleles. Phenotypic expression of alleles are called allelomorphs. One member of each pair is contributed by one pair. When two factors are alternate expression of a trait are brought together by fertilization, the character which expresses itself is called dominant. Tallness condition, which masked is called recessive condition. Dwarfness: the factors are always fewer, and when the gametes are formed, the unit factors aggregate so that each gets one of two alternative factors. It means tallness and dwarfness are separate entities, and in a gamete either TT or T is present. In F1, hybrids are cell-crossed. Two entities separate and then unite independently, forming tall and dwarf plants. Dihybrid cross involves the inheritance of two pairs contrasting characters at the same time. The two pairs of contrasting characters are chosen by Mendel were shape and color of seeds: round yellow seeds and wrinkled green seeds. Mendel crossing pea plants having round yellow seeds with pea plants having wrinkled green glands. Mendel made the following observations.